Huntington's disease, a devastating neurodegenerative disorder, has long puzzled scientists with its delayed onset and relentless progression. A groundbreaking discovery by researchers at the Broad Institute of MIT and Harvard has shed new light on the genetic mechanisms underlying this debilitating disease. The study reveals that the culprit SAG repeat expansions in the Huntington gene behaves like a ticking time bomb, silently accumulating over a person's lifetime. For years, scientists knew that inheriting a mutated HTT gene with an expanded CAG segment was the primary cause of Huntington's. However, the precise relationship between these expansions and the disease's delayed onset remained unclear. This new research has uncovered a crucial threshold. CAG repeat expansions need to reach approximately 150 repeats before triggering the neuronal death responsible for the disease's debilitating symptoms. This finding has significant implications for understanding why Huntington's disease typically manifests between the ages of 30 and 50, despite the genetic mutation being present from birth. The study demonstrates that while these expansions gradually increase throughout life, their growth accelerates significantly after reaching around 80 repeats. This exponential expansion past the critical threshold of 150 repeats marks the point of no return, unleashing a cascade of events leading to neuronal death, particularly in the brain regions crucial for movement, cognition, and behavior. Imagine a delicate ecosystem within the brain where neurons communicate and coordinate complex functions. Now picture a rogue protein born from the excessive CAG repeats in the HTT gene, disrupting this delicate balance. This toxic protein, produced when CAG repeats exceed the critical threshold, sets off a chain reaction of cellular damage, ultimately leading to the death of neurons. The caudate nucleus, a brain region vital for movement and decision-making, becomes a prime target for this toxic assault. As neurons in this area succumb to the relentless attack, the brain's intricate circuitry begins to falter. Like a city losing power grid by grid, the brain's ability to control movement, process information, and regulate emotions gradually deteriorates. This progressive neuronal death manifests as the debilitating symptoms characteristic of Huntington's disease. Involuntary movements, once subtle and infrequent, become more pronounced, evolving from slight twitches to uncontrollable jerks and spasms. Cognitive abilities, once sharp and efficient, begin to dull, impairing memory, judgment, and decision-making. Huntington's disease is a thief, slowly robbing individuals of their physical and mental faculties. The progression of the disease is a heartbreaking journey, typically spanning 10 to 25 years from the onset of symptoms. As the disease progresses, the affected individual experiences a gradual decline in their ability to control their movements, think clearly, and manage their emotions. The once familiar world transforms into a challenging landscape where simple tasks like walking, speaking, and even swallowing become increasingly difficult. The relentless progression of Huntington's disease takes a heavy toll not only on the patient, but also on their families and caregivers. The emotional, physical, and financial burdens associated with caring for a loved one with this debilitating disease can be overwhelming. Currently, there is no cure for Huntington's disease. Available medications can only alleviate some symptoms, providing temporary relief but failing to address the root cause of the problem. This grim reality underscores the urgent need for research breakthroughs that could lead to effective treatments and ultimately a cure. The groundbreaking discovery regarding the critical threshold of CAG repeat expansions in the HTT gene offers a beacon of hope for the development of new therapies for Huntington's disease. By understanding the precise mechanisms that drive the disease's progression, scientists can now focus on developing targeted therapies aimed at halting or slowing down the expansion of these destructive repeats. This new research suggests that instead of solely focusing on managing symptoms or targeting the toxic protein, future treatments could aim to intervene earlier in the disease process. By developing drugs that target the DNA replication machinery responsible for CAG repeat expansions, scientists might be able to delay or even prevent the onset of Huntington's disease. Several pharmaceutical companies are already exploring this promising avenue, investigating potential therapies that could interfere with the expansion process. While initial skepticism greeted the findings, the study's robust methodology involving the analysis of brain tissue and 500,000 cells from individuals with and without Huntington's disease has largely quelled doubts. This landmark research, as many experts believe, has the potential to usher in a new era of Huntington's research, paving the path toward effective treatments and offering renewed hope for patients and their families battling this devastating disease.